I'm not going to be speaking very long. I am Roland Wall. I'm the Senior Director for Environmental Initiatives at the Academy of Natural Sciences of Drexel University. Among other things, that means I help coordinate our work on the Delaware River Watershed Initiative. I'll introduce my colleagues in a moment. I'm assuming most of you know what the Delaware River Watershed Initiative is. It's something that it's been around for a few years. I know some of you I recognize as being involved in it. So, but it is, we've been trying to come up with the 1-minute description, which actually I just sort of redid a little myself today. It's the multifaceted, multi-state, multi-million-dollar project being led by the William Penn Foundation, involving over 50 organizations in working to integrate restoration, protection, and monitoring of watersheds to bring about higher water quality in the Delaware Basin. Now, that's not the official version. Claire doesn't have to report me for not using the script, but that's really, in my mind, that summarizes what we're trying to accomplish here. The Academy has been involved with it since 2013. William Penn has always made has always been a strong emphasis on having a scientific backbone, as we say, to the project, and to making the monitoring work particularly as important as not as important, but certainly as emphasized in some ways as the restoration and protection. We work on trying to understand the conditions of the basin, the current conditions, and also on trying to understand what the impacts are of the various restoration and protection projects that are being funded. Today we're going to talk a little bit about biological indicators. Biological indicators, I'm sure most of you are familiar with, are important ways of measuring water quality and watershed function. It's an important part of what the DRWI is doing. It's been a signature role for the Academy for many years. So we're very happy that the project, the initiative, has given the emphasis to the use of understanding the biology of a watershed before we go. So let me just get started with my colleagues. And so we're giving you 3 mini presentations today, so rather than one long one. And we'll have hopefully some time at the end for questions if people have any thoughts they want to share. So Catherine Christopher, who's going to be our first person up, is one of our staff scientists. She acts as our primary outreach person to the clusters, and if you don't know what a cluster is, she'll explain that to you, but if you're involved with the DRWI, it doesn't need to be explained at all. Greg Barron is also one of our staff scientists. He specializes particularly in GIS and database work, and as well as field work and ecology and a bunch of other stuff I'm not sure on all the time that is very important to what we do, and he'll also be talking a little bit about some of the end user data that we've gotten and some of the algae data in particular, which has been important. And Scott Haig is our senior manager for database and digital products. His team, including Greg, are the ones who try to keep all this stuff organized and make sense of it and help us make some good interpretations of the work that's going on. So that's enough for me. I'll turn it over to Catherine.
Thank you, Roland. Like Roland mentioned, I'm Catherine Christopher. I work with our cluster partners in monitoring within the Delaware River Watershed Initiative very closely. And I'm just going to give a general overview of the monitoring effort for the DRWI. So this map probably looks familiar to many of you. The basin at the inception of the DRWI was analyzed at the HUC 12 level for the watersheds. That's over 400 very small watersheds. And the general stressors that face the basin were identified as forest fragmentation and loss, agricultural runoff, stormwater, and aquifer depletion. And so they evaluated 400 HUC 12 level watersheds for each of these stressors and prioritized them as either being a high priority for conservation where water quality was already good, or priority for restoration where water quality was poor. And these 8 sub-watershed clusters are the areas of high priority where it was found that the overall DRWI effort could have the most impact. So the questions that fuel the monitoring effort in the DRWI are how in-stream ecosystems respond to the on-the-ground actions. And to answer that, we look at which indicators are responding to current stressors and conditions, and how they respond to changes in water and ecosystem quality over time. And we also want to look at how those results can inform the future work of the DRWI and in other initiatives as well. So historically, restoration efforts have not had a lot of scientific input, either before or after the work has been done. The kind of input that it would inform what kind of outcomes you expect to see, or where things could be strategically, where work could be strategically located. But in the contrast to that, the DRWI is very scientific based, and that's what makes it unique. So in general, our monitoring strategy, in order to sort of plan things out, we need to know what we currently know. And what is known widely are the impacts of individual efforts, individual BMPs. We know what they do for runoff, we know what you expect to see in nutrient reduction, et cetera, but there's really only a handful of studies that look at the whole ecosystem response to these works, and especially when there's a lot of different actions being taken place, sort of compounded, what the compound effects are. So there's a need for a larger data set over a longer period of time. And why we need to know the ecosystem response as opposed to just looking at sediment and nutrients, it's really important to look at the biota. In some cases, a stream might be physically intact, or even, chemistry-wise might be considered healthy, but if the biota haven't returned, then they're not performing their functions of nutrient assimilation, migrating the nutrients through the food chain, primary productivity, et cetera. And if you don't have a healthy food chain, as we all learned in 5th grade probably, if you don't have a healthy food chain, you don't have a healthy ecosystem. So our objectives are to assess the fine-scale changes in the ecosystems. And to do that, we look at our changes in the short-term and the long-term time frame by using the methods that we use and our indicators, which Greg's going to talk about later, to their maximum capacity. We are also looking to get a consistent data set with known quality of data. And a long-term, very broad data set, which is what we're gathering, is hopefully, we intend for it to be used as a resource to answer questions in general about ecological conditions and processes. And we look at the whole picture first, and that's sort of where the monitoring effort started out in 2013, looking at what we call integrative sites, which were located to sort of characterize the general conditions of those 8 subwatershed clusters. And then moving forward, we refined the indicators for answering specific questions during future sampling events. In addition to chemistry, the integrated groups that we look at are algae, macroinvertebrates, and fish. We also do habitat assessments. We look at physical habitat as well. And we have some research projects, which I'll only mention very briefly. So who is doing all of this work? The Academy is the science lead for the DRWI. We help strategize. We work with our cluster partners to develop monitoring plans. We provide support, and we provide analysis and reporting. But we also, and just to touch on our history, we've been doing this kind of work at the Academy for nearly 70 years. This is a photo of some early Patrick Center scientists surveying the Conestoga River basin in 1949. And the Patrick Center's namesake is Dr. Ruth Patrick. Her work on diatoms was very important. She discovered that they can be used as responsive and very reliable indicators of water quality, and led to her theory that understanding the biological diversity of an ecosystem is really important to understanding its problems and how to solve them. And the DRWI monitoring effort really stays true to that history, and it's a very multidisciplinary, multifaceted approach. We're looking at whole ecosystem health, like I said. Algae specifically is a specialty of the Patrick Center. A lot of the protocols and methods that Dr. Patrick developed and initiated are still in use by us, by other partner groups, by federal agencies. And that work continues to this day. Some of our most recent research involves developing metrics for using diatoms as indicators of, or how let's see, like an IBI. It's not really an IBI, but it's a multi-metric index for using the algal assemblages in a certain area to quantify the quality of the water. So Greg's going to explain that much more eloquently than I just tried to. In addition to us, we work very closely with a lot of organizations. Specifically, we work closely with Stroud Water Research Center, but this is kind of hopefully everybody else in the basin, sort of broken out by cluster. These are a lot of our partner organizations. And the citizen science effort, which I have in the middle there, is a large part of the data collection effort. So with all of these groups, you can imagine how much data is being collected and housed in our database at the Academy. Since there's so much of it, and we have very specific needs for the data and uses for it, we devised a 3-tier system of sort of categorizing it. This first tier is generally the Academy and Stroud and some other professionals. It's used for the more rigorous scientific reporting, things like that. The second tier is going to be where most of our monitoring partner groups fall. This is going to be trained volunteers who are performing some kind of QA/QC measure, and these data are used to look on a broader watershed scale to identify trends. And the third tier is sort of the everybody else. There's such a wide number of volunteer groups, and they have large data sets. Some groups have been collecting data since long before DRWI was around, and it's really valuable to get that kind of coverage because it helps us sort of flag areas that might need further attention, and it's just really valued for its broad coverage over space and time. So this is a map of our of the basin again, and it indicates the sampling sites. I believe this was from 2014. So this is our sites and the sites that our monitoring partners sampled. But this is just a quick tally. So the of what we've done from 2013 to 2015. The integrative sites are the sort of baseline sites that I mentioned earlier. There's 35 of those. We have sampled the biota there in 2013 and in 2015. So that's algae, fish, and macroinvertebrates. But we return to those sites quarterly every year to take water chemistry and stream flow data. The project sites are the ones that are specifically related to an on-the-ground action. So we would go in and monitor, upstream and downstream of a restoration project or a conservation site. And we have 57 of those just that the Academy has monitored. And then we have our own sort of research questions that we're looking at, individual projects. And those sites, we have 74. So just kind of a quick tally. From 2013 to 2015, the Academy alone has conducted over 1,000 sampling events within the DRWI, including the standard DRWI metrics and our research questions. But if you add to that the over 200 partner group sites that are visited repeatedly, just kind of compounds just how much data there is. So there's a lot. And this is just a list of the taxa that we've collected. Just from the Academy again, it's 275 algal taxa, 46 species of fish, and 347 macroinvertebrate taxa. And this goes through 2015. So those are just some quick numbers to give you a handle on just how much we've gotten. And I'll throw it to Greg now, actually, to talk some more about the algae specifically. So thanks.
All right. So I'm going to get right into the bioindicators. Now, a bioindicator is an organism that shows changes in response to both natural and man-made disturbances. So this could be something physical, like up here in this picture, heavy sedimentation that occurs after a storm event, which can bury a lot of valuable habitat, or something chemical, like having agriculture upstream and the associated high levels of nutrients that are getting into the water because of the agriculture. And what makes an organism a useful bioindicator is if its presence tells you something about the environment. So this could be, for example, if you observe a community of a lot of nutrient-tolerant taxa, you can make a prediction that that water probably has high nutrients. Likewise, if you find a bunch of nutrient-intolerant taxa, you could assume that water has low nutrients. We could also calculate indices of biological integrity, or IBIs, as Catherine mentioned. And these take into account several different metrics. You can look at species diversity, the number of rare taxa, the number of organisms of a certain tolerance level, and it calculates them and sums them up into one score, which you could use to compare between different sites. And it gives you an overall estimation of water quality. Bioindicators are often used to answer questions about pollutant loads, nutrient loads, habitat quality, and why they're very useful in answering these sort of questions is because they capture a longer term evaluation of the water quality. And what I mean by long term is it's like comparing it to something like water chemistry, where you're going out, collecting a water sample, bringing it back to the lab and analyzing it. You're only getting what's going on in the water at that point in time. But when you use bioindicators, those organisms are in there all the time, so they're experiencing any sort of fluctuations or disturbance that are going on in the water, and those communities will reflect those changes. So here's an example to illustrate that point. A simple cartoon. We have a wastewater treatment facility, and we want to see what effect that's having on water quality downstream. So we're going to be sampling downstream for chemistry and bioindicators. First, looking at chemistry, suppose we're testing for total nitrogen, total phosphorus, and total suspended solids. When that water treatment facility is in full operation, you'd expect to have high values for all of these parameters. If we sample again later, and for whatever reason that water treatment facility is in reduced operation or it's not in operation, we'd expect to see much lower TN, TP, and TSS. So there's a temporal variation in this chemistry, so it depends on when you sample what results you're going to get. However, with bioindicators, during its first sampling event, there's lots of nutrients in the stream. You're going to get these nutrient-tolerant, in this case, macroinvertebrates, for example. If you go back and sample later, perhaps the next day, you're going to see a same or similar community, just because those organisms are undergoing every disturbance and fluctuation that's going on in the water. So they're going to reflect those changes. As part of the DRWI, we mainly use 3 different bioindicators: fish, algae, and macroinvertebrates. And for today's talk, I'm going to focus primarily on using diatoms. Now, I guess I should also say we're starting to use or look into using salamanders as well. With diatoms, they're excellent bioindicators because they show rapid responses to changes in water quality due to some of their biological characteristics. They have short generation times, they reproduce, or they grow rapidly, and they just have high dispersal rates. So for today's talk, I'm going to focus primarily on these 2 clusters, the Middle Schuylkill and Schuylkill Highlands, and these are just 2 of the clusters we sample in. The Middle Schuylkill has an area of about 1,900 square kilometers. It's predominantly agriculture at 44%, followed by forest at 35%, and then urban at 18%. And the Schuylkill Highlands is about a quarter of the size at 441 square kilometers. This cluster is mostly forest at 49%, followed by agriculture at 25%, and urban at 22%. So often what we do when we're first looking at the data is we do something like this ordination, which is just an exploratory analysis. So what it does is it takes a multi-dimensional space and projects it into 2 dimensions so you can see general patterns in the data. In this case, this is a canonical correspondence analysis, and it's made up of 2 different matrices. The first one is raw count data that's been log transformed for the diatom data by site. So each of these circles in here represents a different site and the diatom communities found there. And the other matrix is, in this particular case, habitat and chemistry data. Now, having both of those in this one ordination allows us to see how the diatom community changes along an environmental gradient. So we have these 3, Poconos, Kittatinny, Schuylkill Highlands, Upper Lehigh, grouped into a preservation group. And the rest of the sites, the Middle Schuylkill, Brandywine, Christina, upstream suburban Philadelphia, and Jersey Highlands, are in the restoration group. And this is based on land use. So preservation is mostly forested. Restoration, there's a lot of urban and agricultural areas. Now, based on well, each of these circles, so this green circle is for Schuylkill Highlands, pink circle is for the Middle Schuylkill. But just looking at the colors, the green, which are the preservation sites, all tend to fall off to the right side of the ordination, whereas the restoration sites all fall off towards the left. And this is being driven by these different parameters. So there was about 20 different parameters in this habitat and chemistry data set. These 9 here were the most important. So, for example, this arrow with bedrock is pointing up in this direction, which means up here is going to be the greatest percent bedrock, down here is the lowest percent bedrock, and the length of the arrow indicates how much pull it has in that direction. So over here in the restoration group, we're going to have greater embeddedness, greater wetted width, greater calcium, greater specific conductivity, greater pH, higher N to P ratio, and that's driving this the diatom community in the restoration group over in this direction. Next, looking at the same thing, but the second matrix is nutrients in this case. So you see the same sort of general pattern where the preservation sites in the green are going off to the right, restoration sites are coming off to the left. There is a lot of overlap seen with the Middle Schuylkill and the Schuylkill Highlands. So the Middle Schuylkill is this entire area in pink, and the Schuylkill Highlands is just in green. There's a lot of overlap. So based on that, we predict that they're being influenced by these variables in a similar way. So they probably have high or similar TN, similar TP, similar potassium, similar sulfate, just on based on where we're seeing these diatom communities in this ordination. And we can look at that further by using these TP and TN metrics, which were developed at the Academy. These are based solely on the diatom communities and known tolerance values for the species that have been observed. So a score of 0 means, in this case, low TP. Score of 10 means high total phosphorus. Here we have the Kirkwood-Cohansey and upstream suburban Philadelphia included, but we only have one site that was sampled in this particular year, so you could ignore those for now. So over here are the restoration sites. On the far side in the green are the preservation sites. So you can see there's a pretty clear distinction for the most part in TN and TP. So TP and TN are higher in restoration, lower in the preservation, with the exception of Schuylkill Highlands, which is much more similar to the restoration sites, which we could have predicted from that past ordination where there was a lot of overlap seen. This is another metric that was developed at the Academy, and this is a multi-metric index. This has about 124 different metrics used to calculate this one score. And depending on where you are in the United States, you're going to use a different subset of those metrics to get the score. And this gives you an indication of overall water quality. So a score of 0 means the worst water quality, score of 10, best water quality. Now, again, we do see preservation sites have better water quality, which we had guessed based on the land use. The Schuylkill Highlands has significantly better water quality than the Middle Schuylkill. And if you remember in the past figure when I showed TP and TN, they were fairly similar, which indicates that something like nutrients or water chemistry are probably more important than nutrients and influencing the diatom communities in these two clusters. And this is showing the same thing, but to help you visualize it, it's projected on a map. So each of these is a cluster, and the circle represents the overall water quality based on the MMI score. So good means, or green means good, yellow means fair, and red means poor, and the size of the circle represents the number of species seen. So down here, where it's very urban, upstream suburban Philadelphia, it's got poor diatom community, or poor water quality based on the diatom community. Brandywine Castrina is similar. Middle Schuylkill and Schuylkill Highlands, based on how this is broken down, both have fair water quality in regards to the diatom community seen there. So in conclusion, for the bioindicators portion of this talk, both the Schuylkill Highlands and the Middle Schuylkill have high TP and TN according to those algae TN TP metrics. However, the Schuylkill Highlands has significantly better water quality according to the MMI scores, which suggests that nutrients and water chemistry are probably more important in influencing those communities. And finally, the clusters with less agriculture and urban areas tend to have better water quality according to algae. Next, I'm going to turn it over to Scott to talk about the database.
Just real fast, I
Yeah.
should have said this at the beginning. If anyone has questions, feel free to dive in. Otherwise, we'll have some more time for questions at the end. So, anyone has anything immediate for Greg while it's still in your mind? We're getting a little technical here.
Yeah.
Do you guys have some kind of tutorial or guide to using diatoms for bioindicators?
There's published papers that have been done at the academy, but I'm not sure if there's like
I want to use something with my students.
Right.
We
I'm not.
follow the
Is there?
swamp protocol, which was developed in California,
Okay.
and we sort of do a revised version of that. But you can find it online if you just, SWAMP is an acronym for something.
I'm sure it is.
Yeah.
Okay.
But it was, like I said, it was developed in California, but we,
Tweak it a little bit.
yeah.
I mean, we have a ton of material on diatoms. And
I know you do.
about it.
And
feel free
I can
to,
use that class.
yeah, if one of us will give you a card, you can reach out. We'll
Okay.
hook you up with our phycology people. But there may be some stuff. We do a lot of diatom work on the National Water Quality Assessment, NAQUA, for the USGS, and there may be some explanatory material with that one as well.
Okay.
So.
Yeah.
If you're focusing on diatoms, is silica ever a consideration? Because it's a very important nutrient but is it never limiting for diatoms in flowing streams or?
Well, in that original habitat and chemistry data set, it was in there, but that wasn't one of the parameters that was selected as being the most important based on, with the canonical correspondence analysis, it also runs a multiple regression and picks out the most important variables, and that just didn't happen to be one, which probably means it's not limiting in these systems, but I'm not really sure. Yeah.
I have like a sheet of all the taxa of macroinvertebrates, and if it's a 0 to 10 or whatever, 0 to 9 scale for how pollution tolerant it is.
Mm-hmm.
Is there something that lays out for all of those taxa in eastern US or something, what they would be more sensitive to? So, you know, if I'm finding a certain group, is there like a database list of that somewhere that anybody's aware of?
Like for
versus-
macroinvertebrates?
Yeah, versus, you know, looking each one up individually, let's say.
Right. I'm not sure off the top of my head, but I would assume there's one.
Yeah, I mean, there's different, the way you use macroinvertebrates is partly based on their tolerance and partly based on the kinds of indices that you're looking for. So I think if you're trying to parse out if this is an issue with DO or is this an issue of too much nutrients, it can be, you know, in part because of the individual taxa, but it may also be the set of metrics you're using together. So that may, you know, I again, speaking a little out of my area here, but that would be my thought. Preston, you've been in this business for a long time. Any thoughts on that? I saw you moving the water.
Yeah, I mean, I had a PhD working for me for a while at Aqua who was focused on just looking at all of the different metrics and ways of looking at these things. And, you know, every day he'd come in with a new preferred method and a preferred idea and what they really meant. When you're looking at biodiversity of macroinvertebrates, sometimes flow regimes are important, and it has nothing to do with nutrients or anything else.
Right.
You know.
So, yeah, so there's a lot of different ways of skinning the cat on this and measuring, you know, what you think you're going to get out of it, depending on the particular species. And then there's others that are more just commonsensical in terms of if you've got a lot of these, you've probably got a problem, or if you don't, you may not, you know. I mean, it's the reason we use things like EPT and chironomic counts as being sort of rough indicators of where things are playing out. So. Any other questions for now?
All right, Scott.
All right, now for the most fun part of the talk. So I'm going to talk about the database that we're putting together as part of the DRWI project. I started out my career doing a lot of field work, and then I got into GIS and remote sensing, and then into information systems. And I'm now at the Drexel PhD program at the I School, what's now called CCI. And so I just, to give a little perspective here, in the environmental science, looking at the environmental world, visualizing and analyzing the world, there's only one world. So if you want to do any kind of experiments, if you want to do any kind of changing the world, it has to be done within the computer system, within models. And so early on, in a lot of my conversations with Roland, we talked about how we would integrate all this information that was coming in for the DRWI. And just to give you some perspective of that, we can talk about just information, environmental information collected by different government agencies. And you can spend years putting all of this information together into one place. I know Greg does this all day long. You have the DEP data where they have the land use data, say for New Jersey, and all the information on the toxic release information from EPA. USGS information on the real-time stream gauging data, or the soils data, or the aquifer data. We in Penn paid Shippensburg University to put out a 1-meter land use land cover data set for the entire watershed. And so some of the informatics perspective is that's 900 times bigger than the NLCD. It's 30-meter NLCD versus a 1-meter land use land cover data set. So how do we take that information and bring it and make it useful? It's so much bigger. It's terabytes and terabytes in size. DRBC data, or even the Army Corps information on dams. So we have all of these different perspectives on types of information, and all different ways on how they're collected, from remote sensing products to GPS systems to people out in the field collecting information. So that's the background of data. And if you talk to environmental scientists at any university, they'll talk about how they integrate this data, and they can get a new perspective on the world, and they can make decisions on it. And so that's been some of our issues within the DRWI, is bring all of this information into one system so we can look at it at one time and ask our questions in a unified way. And then on top of that, we want to bring in all the information that our partners and the people within the academy are collecting and bring that into the system as well. So this is a very big informatics and computer science problem, which is what really interests me. This is sort of what I'm really excited about. So, our first thought about that is, where do we store all the data? And we looked around at different techniques to store large data sets like this. We're talking about terabytes of data. We're talking about remotely sensed imagery. We're talking about LiDAR, laser collected data, satellite imagery, imagery from all the major agencies, information from the academy, which goes back hundreds of years. And the question is, where do we put all of that, and how do we store it all? And we decided we wanted to use a free relational database management system called Postgres, which is the largest relational database management system, free one in the world. There's a couple of reasons why we chose that. One, we want to make sure any partners who want to work with us can get it, and they don't have to pay a lot of money. The companies that sell relational database systems like Microsoft and Oracle, they can cost $15,000 to $20,000 a year for a single license. And this is free, like I said, so anybody can use it, they can interact with us, they can download it and process it for free. Second of all, it's really powerful. It has spatial data processing internally inside of it. So if you're familiar with how relational database systems work, they're really matrices of data that you can compare based on keys of information, how you relate one table to another. What's really exciting about this relational database system is it can relate data based on location. How far away is something from something else? Is it up or downstream of something? How far away is it up or downstream? How long would it take to travel the stream to get there? These are all questions you can ask about data within your relational database system. And then we thought about periphery software that we'd use to interact with the system. How would you put data in? How would you create some of those graphics that Greg showed from the data set? And we used open source software packages. R is a free statistical software package here. We have Quantum GIS, which is a free GIS package. So if anybody wants to interact with our database, they can get access to it for free using mapping tools like this. And we picked a front-end tool called Django, which is a Python-based front-end web system. The only pay software we use is Microsoft Access to enter form information, so people can directly enter information into the system. There's one slide there that was missing. One last thing I'll say about this is this whole process, getting information into the system in the database terminology is called the extract, transfer, and load, or ETL. That's a pretty common industry standard. Relational database systems were invented in the 60s by the idea of relational database theory by someone named Codd. And the idea is that information should never be stored inside your system more than once. So this idea allows us to simplify data. For instance, if we have one sampling location, we never store the location more than once, but we may store hundreds of columns of information about that location, and then just join them together by a unique field. That allows us to minimize the amount of times we store the spatial location of the data. And a lot of database problems, if you've done a lot, I've done this work for 14 years now, integrating data from other agencies, a lot of the database issues you'll find with their data is simply because they don't bother to follow the normal tenets of database construction. Normalization, duplication of data, numbers that are clearly out of bounds. Greg and I have spent weeks dealing with the NIPD's data set from EPA, and there are just some things that you wonder about how they got in there, because they're saying there's 200,000 million gallons of water coming from a wastewater treatment plant during a day. Is that possible? And there's questions like that. Well, within our database, we're creating all kinds of rules that say the data, you couldn't expect a number like that to exist because it's too high. You couldn't have a sampling site be collected twice on the same day, or even twice within a couple of weeks of each other, because that would break our rule we have for the data. So we've been spending a lot of time building our standardization and our QA/QC process within the data. And now the database has gotten so large we can really start to ask some interesting questions, and that's the next step of our process. We've brought so much information in, we've integrated it through this ETL process, we've normalized it, we've projected it into the exact same GIS projection system. We're using UTM projection system, so all of our spatial information is in the same way. All of our temporal information is in UTC time, so we can ask questions about time. Everything's the same across all the agency data, across the academy data, and across our partners' data. So we can now ask these questions without having to spend so much time cleaning the data up anymore. And so what we're really trying to do is empower the scientists that work on this project so they can ask more and know more about the data. So I wanted to talk about one way that we're using all of this information. This is just one example of how the data is going to be used and has been used. We're working on a phase 2 planning tool for the DRWI. And this is work that's happening at the Academy, but it's also happening at the Open Space Institute and the National Fish and Wildlife Foundation, as well as with Dr. Barry Evans, who's with Penn State University, and also with Stroud Water Research Center. We're all working together on this tool. And this is just an example of the NIPD's data set I was talking about before, the National Pollution Discharge Elimination System data set. This area of the basin has roughly 1,188 data points, and one of the challenges that we've had is that we get this data from EPA. We also can get this data from the PA DEP and the New Jersey DEP, and they don't agree. So that's a huge challenge. There's like 80% difference between the state agencies and the federal agencies on the database. So that's been we've had a lot of issues. Just the state database is built, and then it is passed on to the federal government, which then builds this database. So we still are trying to figure out exactly why they're different and why they're showing different types of information. But
I'm sorry, is that the scope of Watershed?
It is, yeah. Yep. And you can see that it's broken down into these different types of facilities. These are all point sources of pollution across the watershed. And you can see that most of them are privately owned, or not quite half of them are privately owned. We have corporation, municipality, water treatment, unknown, state government, mixed ownerships. These are all the different types of facilities. And one of the things that we're interested in for the DRWI is if you're planning to do a restoration or a preservation project, what's upstream of where you're planning to do the work, and what potential impact can you actually have given what's above you? And there's a couple of perspectives on that. One is all of these facilities are releasing a certain amount of effluent with a certain amount of pollutant, but they're also permitted to release a certain amount of effluent and pollutant. So we can look at both of those things, and we can model, and I'll show an example of this, for each stream breach, what's above it and how much pollution is being released above it. So that's the point sources. And then the non-point sources, we're working with a model called MapSheds, which was created at Penn State by Dr. Evans. And we're using his model to allocate TN, TP, and TSS loading by land use cover type and soil type and rain flow to all the different watersheds within, again, the stream area. So these are the NHD Plus version 2 watersheds. There's roughly 15,000 of these across the Delaware River Basin. And we can allocate to each one a predicted TSS, TN, and TP loading based on the land cover. And again, we can then allocate that down the stream and predict the total loading upstream for every reach within the watershed. So the idea is how much loading is happening at one place, how much of that loading can actually be addressed with a BMP or with a preservation project. And so to give you an example of that, this work that Dr. Evans did predicted for a Huck 12, which is a larger unit aggregation, a watershed, there are 400 some Huck 12s, there's 15,000 of these smaller reaches. His work predicted here what the loading would be at the bottom of the watershed. And what we've done working with him at the academy is we've redistributed that load based on the land cover out across the watershed, all the way to the top of the watershed down to the Delaware Bay. And then once we've predicted at each watershed what the loading would be, we then we can route it down the stream. And as we route it down the stream, we're summing the cumulative loading. So you can see that the amount gets bigger and bigger. And then as we reach these point sources of pollution from the NIPD's database, we're then adding that in, the estimates of the TN and TP loading from there. And so that then goes all the way down to the watershed. You can see, again, it's a cumulative loading. And the idea is that we're going to be working with scientists from the academy, from Stroud, to look at what the impact of this loading is on the ecological communities based on some of the data that Greg showed, and then to think about how much effort would it be required for each of these reaches to get the numbers down to an area that you would expect an ecological change to happen. So the question is, what's the order of magnitude to change these streams versus these streams? And what would you have to do to see a change in the biological communities in the streams? So that's where we're trying to connect the modelers, the people that are doing the work on the estimates in the watershed to the ecological perspective, and then trying to take that back to the NGOs and their work, and trying to inform them about where maybe the best place to do the BMPs may be. And so some of the information you'll get, we can get information about the reach, the watershed here, it's percent developed, percent ag, percent forest, its average TN loading in its area, but then also, everything above it. So what's everything that's draining into here? And what's our ability to change all of this above it so we can actually see a difference in stream here? And then that's the phase 2 planning tool, and that work should be done sometime in July, so we're still working on this. The other perspective is all of those data sets are going to be available internally, not just for the phase 2 tool, but for internally within the academy, and if others are interested in looking at some of the data, we'd be glad to talk with you about that. And lastly, just to sort of end the time here, one of the things I'm seeing more and more of in working with other scientists at Drexel is this idea of data mining and pulling information out. So I just ran this through the NIPD's data set, just looked for a keyword, and you can see some of the keywords that come out of NIPD's. It's kind of interesting, actually, that MS4 shows up very high here, which is something that we're going to need to think a lot more about how that's going to influence some of the water quality as that process continues. But you have sewage treatment plants, boroughs, and then you get down into sort of the weeds where the towns, birds burrow and sinking. So I think there's going to be, as the data becomes more digitized, as information moves from books and from physical manifestations into the digital world, you're going to see more machine learning algorithms, you're going to see more types of ways to understand the data because it's a very complicated data set. There's so much information in there, it's very difficult for a single person to understand it all. And so we're starting to rely more and more on sort of the machine learning approach to thinking about how we can extract information from this to make more informed decisions. So that's my talk about the database. I guess I'll take specific questions about the database, and if there aren't any of them, we can take more general questions about the work.
One thing I'd just add to what Scott was saying, and he alluded to, is that this sort of closing the loop here between the amount of nutrient loadings, its impact on the watershed, what we can model in terms of those amounts, and then what the ecological outcomes are, and well, first of all, the water quality outcomes are, and then how that reflects in ecological metrics, is really sort of it's a bit of a holy grail, I guess, in terms of what we're trying to do here, because it's data that, remarkably, there's not a lot of specifics about, okay, if I put this BMP in this place, it's going to have this effect on loading, which will in turn have this effect on water quality and ecological outcomes. But, going all the way back to the algae or the macroinvertebrates or whatever it is that we're using as the indicator. There's pieces of that puzzle all at different places. There's certainly, things that get associated with BMPs according to nutrient attenuation, and there's things according to how much nutrients affect ecology. But trying to put this in, particularly for a specific set of watersheds like the Delaware, is really one of the long-term, I think, aspirational goals of what we're doing with the science here, so.
Yeah, I would just add to that perspective, at least in DRWI, has happened because the DRWI has made a lot of different scientists work together. So on the modeling side, you have people from Penn State and Temple and Villanova that have heavily focused on nutrient modeling. And on the academy side, we have a lot of ecological data. So it's this unique perspective where we're bringing together some of the modelers with some of the ecologists that we get this different perspective, this different point of view on how they can relate to each other. And then the other thing I think that is useful is we've started to really look into technologies that allow us to understand what's up and downstream of a particular reach very fast. So we've developed tools to do graph theoretic tools that allow us to route the watershed very fast, which gives us also another perspective.
And we and our partners at Stroud are going to be assembling a group later this spring of key ecologists to talk about the very specific ecological outcomes that might be found. So that work group is going to meet a couple times and try to help build up on the science of this. And as Scott said, the DRWI, it's really been one of the, I don't know if it was one of the predicted outcomes, but it's certainly been one of the, from the scientific standpoint, one of the major outcomes is you have these collaborations of people, you have opportunities, even something as concrete as looking at salamanders, which is something that our team's been working on now for a couple of years. They're a useful indicator. They can be used in places where fish populations may not be as accessible. And so, but there hadn't up to now been a lot of sort of indices that directly relate to salamander. So all of these are sort of follow-alongs that come with the really important restoration protection work that's being done. So that's the question.
Yeah.
Question, I love that last little model and how this predictive element. My question is, how close to the site-specific kind of information on land use and land cover can you get? Did I hear you had some LiDAR kind of pieces mixed in this predictive model?
Sure, I can answer that. There's sort of two answers to that. One, the data is always getting better. So we're getting more and more information all the time. So the land use data that the original MapSheds model that was run on was based on the NLCD 2011. It's a National Land Cover data set. It's a 30-meter data set created by USGS and NOAA in a joint work based on Landsat satellite imagery. So it's every 30 meters on the ground, they can give you an estimate of whether it's urban or forested or agriculture. So that they're sort of major large groups. Now, what's going to be coming out soon is this much higher resolution spatial data set from Shippensburg, but we don't have that right now. So I would imagine that, like everything else, these things will be updated with that newer information. But that will be at 1 meter, and that used a fused LiDAR and aerial photography fused together, and it's much more high resolution. So if you imagine a 30-meter pixel, it may cover multiple land cover types. There may be a little bit of forest on the edge and a parking lot and some grass, whereas this 1 meter data set will bring them all apart. So it would be much higher resolution.
But as Scott said, it's also much more data intensive.
Yeah, it's 900 times more intensive. But it's also in some situations older, because the LiDAR data was collected in 2006, where NLCD was 2011. So there's always this temporal spatial trade-off with these data sets. Oh.
So I have two questions. One, I guess you mentioned if with an OSI.
Mm-hmm.
So is there a discussion of how that might influence their funding as far as conservation projects or restoration projects? I'm sure that's probably going to come down later. Or
yeah, that's a question. You're probably asking the wrong person that question.
right.
I'm building the model.
Yeah.
So.
I think the bottom line is probably yes. I mean,
Okay.
so the goal is to have the stream reach tool, which Scott was talking about, be the basis for an improved criteria for OSI and FWIP projects
Okay.
going forward, but we're not there yet.
all right.
We do, from my perspective, I do see that Barry and I will be meeting with a lot of the different cluster groups to talk about how accurate this is and what their perspectives are. So there's some estimates we have to make necessarily because we're looking across 15,000 square miles, like where farm animals are. And there may be much more accurate information of the clusters that we would like to integrate to make this a better perspective.
This is absolutely phenomenal.
Oh,
I mean, it
thanks.
really sheds a lot of light on what we've been hearing, we're kind of leading to do, but this is like, we get to see some of the results of it here.
Well, it's been in the works for the last couple of years, and finally, some of the stuff is starting to come into land.
I mean, it's
so
really,
it's coming together slowly.
So a question about how you developed this, what you did the word search on, was it for the entire Delaware Basin, was it newspaper articles, or just the NPDES permits, or?
It was on the, this was just on the NPDES permits, and I believe it was just on the Schuylkill Basin, but I could get back to you exactly. It was just something I wanted to throw in as far as this
Yeah, okay.
data
I mean,
searching idea.
I see Abington and Malvern in there, which, you know, so I'm thinking maybe it wasn't the.
Maybe it was the whole NPDES,
But,
yeah.
but whatever. If you could go back about 3 or 4 slides, there was one, yeah, no, keep going. Keep going. There was one about, no.
Oh, the graphics take a bunch of slides. Hold on.
Yeah, that one and the one before it. Okay, the red and the green segments there, are those impaired and non-impaired stream segments, or what are the colors on the stream
Right.
segments?
Yeah, the colors are just the aggregation of the nitrogen. This is TN estimates, but I didn't want to really get into the, I mean, I didn't mean it to talk about it in that way. I mean, it's really just to show that as you get downstream from these point sources, that you're getting a higher amount of loading of nitrogen. So the idea is that up here, you're having this point, but by the time you get to here, you've got 1,188 points. So it's just a visual description. Does that make sense? There's no
The color
exact.
of the stream segments are a representation of what the theoretical loading
Yeah.
might be in the stream, or concentration might be.
That's right. Concentration's a whole different thing, but loading.
Okay,
Yeah.
all right.
So is that your question?
Some of the questions, Patrick, you said you had 2 questions? or-
Oh, well, I kind of talked to Catherine earlier about this, but I was trying to get a timeline for the salamander
Oh.
methods. Is there any?
I think there's a paper in the works
Yeah,
that's
that's
gonna
what I
be
thought.
out at some point. I mean, we could probably get you some insider information at some point if you want
Okay.
to give us a call. I mean, I don't think there's, obviously not a secret about it, but
No.
they're using, I think there was only one existing salamander index around in Ohio or New Jersey or somewhere, and so they're trying to come up with something that's more focused on the Delaware basin, so.
So, can an organization engage you guys or one of your partner groups to kind of help plan BMP implementation? Is that the intent here?
Well, the intent of the tool is to create a tool to think about where BMPs can be put, where they can actually have a change on in the stream for the DRWI. I should be really specific. This is really designed for the Delaware River watershed. It's not a general BMP design. Where would you want to put BMPs for any reason? There's specific things that William Penn is looking for the DRWI that we're incorporating within the tool. But I think it could be, after it's all built and all that work was done, I think there could be a lot of other uses for it. And there's a lot of data there already. There's a lot of algorithms there. So I think it could be co-opted to answer other questions beyond what's built.
Yeah, and I would just add, Claire stepped out, but I'll speak to the foundation to some extent and say, I'm pretty sure they want everything to be publicly accessible, they want everything to be transparent, so if we're developing a tool, it's certainly not gonna be proprietary, it'll be something that people will have ability to make use of. There may be some role for us in helping support that in the future, and certainly, be willing to work with groups on that as well.
I should say that we are developing it for the entire watershed, not just for the clusters.
Right.
So, the whole point was to make it available for anybody who was interested in using it. I'm just saying it might not be exactly, it might not answer the exact questions you're interested in versus what the DRWI's questions are.